Hello, everyone. Welcome to today's session of Biomolecules. And in today's class, we are going to study about the nucleic acids. So, guys, am I audible? First, let me check that. Yes, I am audible. So, I welcome you all to this wonderful, awesome channel called Let's Crack NEET UG in English, where we are continuously striving to provide you best of the best content so that you can utilize it to your maximum and you are able to score a really good rank in NEET UG examination. So, guys, in this series of lectures, you're regularly getting live lectures of all the three subjects physics, chemistry, and biology in your favorite channel, which is Let's Crack NEET UG in English. Okay, so my name is Aditi Gupta, and I'm your biology mentor. And in today's class, we'll be discussing about the fourth type of biomolecule, which is nucleic acid. We have already discussed the basics of the biomolecules, like how can we isolate them, what are the criteria for its classification. Then we have studied the carbohydrates, proteins, lipids, and now today is the turn for the nucleic acids, right? Although the nucleic acids you will be learning in detail in your class 12 chapter molecular basis of inheritance. I have tried to include all the basic points in today's lecture only, which will definitely help you in understanding when you'll be learning molecular basis of inheritance. Okay, so let's start today's session without any delay. So guys, most of you already know me, so before proceeding for today's session, first let me introduce myself and tell you something really important about Unacademy, and also how can you connect from us. Okay, so basically, this is the Telegram channel link. All the educators of this particular channel are available on this Telegram channel. So those of you who have still not joined, so this is the link. You will be getting all the updates of the upcoming lectures on this Telegram channel. Hi Sajjan, welcome to the session. Now, talking about myself, so students, my name is Aditi Gupta, and I have been mentoring students for the past 6 years for their NEET UG preparation. Okay, many students of mine are in their dream medical college, and trust me, now it's your turn, guys. Okay, all you need to do is to study hard, to be honest to yourself, and to put your honest efforts, right, and do your best. Students, I myself have secured an all India rank of 61 in the prestigious GATE examination and have pursued my masters in biological from IT Kanpur in biological sciences and bioengineering. Okay, sometimes confusion happens. Now, see what happens. There are topmost team of educators from all across the country on this amazing platform called Unacademy. Okay, so students, how can you avail the maximum benefit? How can you avail maximum benefit? So the idea is to get an academy subscription. So the idea is to get an academy subscription. First, let me clear something really important. See, students, we are covering the entire syllabus here. Okay, we are covering the entire syllabus here, and we are that too. The complete NEET UG syllabus will be covered here. That too in a certain time limit also, because we need some ample amount of time to revision. So do not worry about the content. Please go with the flow. Whatever topics are being dealt here, you know go conceptually, read NCRT, practice number of questions. We'll be definitely covering each and every chapter, so do not worry about the syllabus. Okay, guys? Okay, Sajjan, this is for you. Now, I was talking about the subscription part. So first of all, how can you get the subscription? Students, you need to download Unacademy learning app from Play Store or iOS. Okay, now, this Unacademy Learning app has some absolutely free of cost features like these special classes. These are the screenshots of mine special classes. And I take my special classes in, bilingual mode, like Hindi English mix, right? So my timing on Unacademy Learning app is 11 a.m. Okay. Apart from special classes, there is another feature which is absolutely free of cost, which is the quiz features. So, by downloading an Academy Learning app and by exploring these free of cost features, you can enhance your learning experience. So, I suggest you to immediately proceed for the same. Right now, coming towards the benefits of having an Academy subscription, guys, you will be getting live classes from the topmost team of educators, and you can select your batch according to your requirement, and you can track your preparation. Okay, so there are two platforms available on Unacademy subscription platform, Unacademy Plus and Unacademy Iconic. So let me tell you the benefits of having an Academy Plus subscription. Students, you will be getting daily live classes of all the three subjects, physics, chemistry, and biology will be covered in detail by the topmost team of educators. Okay, physics, chemistry, and biology will be covered. Okay, by the topmost team of educators, and you have the flexibility of choosing your educator and your batch depending upon your convenience, your understanding, and your compatibility. Okay, there is no fixed thumb rule. So you have the flexibility here, okay? Then the complete NEET UG syllabus will be covered in a detailed manner, in a thorough manner, along with the detailed PDF notes, and these PDF notes are provided to you directly by the educator, so you don't have to worry about which book to follow. Okay, we are here to provide you each and every updates. Okay, then you will be evaluated on the basis of the live tests and quizzes. There are multiple test sessions, okay, weekly test, or after the completion of a particular topic. So there are multiple plans available, and this is to check your knowledge. Okay, because ultimately you have to perform in the NEET examination, and whatever theory you have learned, you need to learn how to solve questions from them. Okay, and then you will be getting unlimited access to all the NEET UG content. That means there are around 50 plus educators on this platform, okay, on Unacademy subscription. So you don't have to subscribe each and every educator, right? With the single subscription you will be getting unlimited access. That means you'll be getting access to the video lectures of all the educators, their PDF materials, and their test series. Okay guys, so this was about the plus. Now talking about the brand new platform which Unacademy has recently introduced, that is the Iconic platform. So guys, Iconic platform has some out of the box features, like you will be getting personal coach here, and your personal coach is there to listen to you, to listen to your problems, to listen to the challenges which you are facing. Your personal coach will be there to guide you throughout your NEET UG preparation journey. Okay, to he or she will prevent you from deviating from your path, because your path is to clear NEET UG examination, right? Then you will be getting study planner, so that you can utilize your time to the best of your efficiency. Okay, and these study planner will be reviewed with the help of bi-weekly meetings, so that we can see whether the particular study planner is working on you, or we need to do certain improvements or not. Okay, then talking about the next part, which is the test analysis. So you will be provided with in-depth test analysis, so that you can check what mistakes you have done in any particular test you have attempted. Okay, and this is how you will be improving your problem solving skills. Then all the benefits of the plus platform are included here. Okay guys, you will be getting expert motivational sessions from time to time to keep you motivated, to keep you enthusiastic. So this was Unacademy Plus and Iconic. So in both the platforms, you are getting India's best team of educators, interactive live classes, structured courses and PDFs, live tests and quizzes. But what's new in Iconic? Availability of personal coach, study planner, your own personalized study planner, expert guidelines and test analysis. Now let's see how can you proceed for the subscription. So if you have decided to go for Unacademy Plus, so see, there are multiple subscription plans available, but this is my suggestion to you, that the longer the duration of the subscription plan, the more benefits you will be getting in monetary terms, and also in the terms of the education which you are going to receive, because you will be under the guidance of the topmost team of educators for that particular duration, right? And normally 1 to 2 year of time is required for NEET UG preparation. Okay, but those students who are preparing for NEET UG 2021 and have still not bought their subscription, I would suggest you can go for 6 months of subscription plan. Okay, so we have 6 months, 12 months, 24 months. See, the price for 6 months is 28,000. For 1 year it is 38,500, and for 2 year it is 56,000. But if you use my referral code, which is AG001, you will be getting an instant 10% discount on your course fee. And now you have to pay only rupees 25,200 for 6 months, 34,650 for 1 year, and 50,400 for 2 years. And remember guys, always compare the monthly cost which you are paying. Okay, see, the minimum monthly cost you are paying in the 2-year subscription plan, right? Even if you see the monthly cost, 1 month subscription fee is 7,000, and if you buy the subscription for 2 year, you have to pay only rupees 2,100 per month. So only at rupees 2,100 you are getting all the benefits of the plus platform. Similarly, about the Iconic has 3 subscription plans: 6 months, 12 months, and 24 months. 6 months cost you 38,000, 12 months is 65,000, and 2 year is 1 lakh. But again, if you use my referral code AG001, you will be getting an instant 10% discount. That means now you will be getting a discount. Okay, so for 6 months you have to pay rupees 34,200. For 1 year you have to pay 58,500, and for 2 year you have to pay rupees 90,000. Okay guys, and on 2-year subscription of Iconic, you are paying only rupees 3,750 per month, and you are getting your own personal coach, live classes along with regular doubt clearing sessions, complete NEET UG syllabus coverage along with the PDF notes, live tests along with the test analysis, okay, in this particular amount. So do not wait to waste your time. Go buy your subscription and start preparing as soon as possible, okay, because time is crucial, my dear students. Now coming towards our today's topic, guys, today's topic is the nucleic acid, okay. So what are nucleic acids? We'll be starting from the very basic of nucleic acid, okay, and then we will be discussing the type of nucleic acid, okay. So talking about the nucleic acid, how was it discovered and how was the term nucleic acid derived and what is it made up of, okay. So see what happened. It was first discovered by mixture in the nucleus of the pus cell. In the nucleus of the pus cell, white blood cell. Okay. And it was termed as nuclein at that time. When mixture found it in the nucleus of the pus cell, it was termed as nuclein. Okay. Term nucleic acid was given by Altman. So term nucleic acid was introduced by Altman. Okay. And what exactly is nucleic acid? So nucleic acids are the polymers of nucleotides. They are the polymers of nucleotides. Now what are these nucleotides made up of? So the nucleotides are made up of nitrogen bases, pentose sugars and phosphates. Okay, so in short I'll be writing as N2, that means nitrogen base, then pentose sugar. Okay, so I'll be writing simply the sugar and the phosphate ion. So all these three units combined together comprises a nucleotide. But in order to understand the nucleic acid structure in detail, first we need to have a clear-cut understanding of the component of the nucleic acid. Okay, so one by one we will be studying about the various components like the nitrogen base, sugar and the phosphate ion. Okay guys, because this is my teaching style, I want to cover from the very basic so that you yourself can connect each and every dots. Okay, now see, there are two types of available nucleic acids: ribonucleic acid, that is RNA, and deoxyribonucleic acid, that is DNA. And there is a difference in the structure of both these nucleic acids. I have used a single stranded molecule of RNA because RNA generally is present in the single stranded form, whereas DNA is present in the double stranded form. So DNA is represented by the double strands. Okay, now see, the nitrogen bases, which are nitrogenous bases, which are present in the structure of the nucleic acids or in the structure of DNA and RNA, are broadly classified into two categories: purine nitrogen base and the pyrimidine nitrogen base. As you can see, the purine nitrogen base is a bicyclic ring structure. It contains two rings. It is a bicyclic ring, right? One is a 6-membered ring, another one is a 5-membered ring. 6-membered ring is the pyrimidine ring. 6-membered ring, the name of 6-membered ring is the pyrimidine ring, and this pyrimidine ring is also present in the pyrimidine nitrogen base. This 5-membered ring is the imidazole ring. If you'll be studying about these names while studying organic chemistry, so do not worry. Okay, now see, the cyclic structures which are present in the nitrogenous bases, they are basically bicyclic, and the purine is bicyclic, but basically they are heterocyclic compound. They are heterocyclic compound. What is the meaning of heterocyclic? See, along with the carbon atom, nitrogen atoms are also present in the ring structure. When we see the purine, so we can see that at position 1, 3, 7 and 9, nitrogen atom is present. Whereas talking about the pyrimidine, in the structure of pyrimidine also at position 1 and 3, nitrogen is present. Such type of ring structure is known as what? Such type of ring structure is known as heterocyclic ring structure. Okay, now what happens whenever this nitrogenous bases, it combines with sugar. Whenever it combines with sugar, they always attach to the first carbon of the sugar molecule. They always attach to the first carbon of the sugar molecule. Okay, so examples of the purine nitrogenous bases are the adenine and guanine. Adenine and guanine represented by A and G. Okay, so adenine and guanine are the example of purine nitrogen bases. Similarly, the pyrimidine nitrogen bases are cytosine, as shown here, uracil in case of RNA. It is specifically present only in RNA, and thymine in case of DNA. It is specifically found in DNA. Okay, so pyrimidine nitrogenous bases are also of two types: cytosine or uracil in case of RNA, or cytosine and thymine in case of DNA. So this was about the nitrogenous bases. Okay, now let us see the structure and property of the sugar. Okay, so the sugar molecule which is found is basically a pentose sugar. Okay, so this pentose sugar, that means it contains 5 carbon. It contains 5 carbon. Okay, so what happens? The pentose sugar, which is present in case of RNA, is the ribose sugar. As you can see, I have included the structure here. So this is carbon number 1, 2, 3, 4, and the fifth carbon is not included in the ring. It is present outside the ring. Okay, so this is the structure of the ribose sugar, which is present in RNA. The sugar which is found in the DNA molecule is the 2-deoxyribose. Okay, and there is a clear cut difference between the ribose and the 2-deoxyribose. See what happens. You can see the second OH, you can see here the OH group which is present at the second position. Okay, this OH group is not found here in case of the deoxyribose sugar. So it is believed that during the process of evolution, certain structural modification took place in the structure of DNA, which provided more stability to the structure of DNA, and this favored in DNA in behaving as a genetic material. So at the second position, the hydroxyl group is not present in case of 2-deoxyribose. So these are the type of sugar which is found in the structure of nucleic acid. Now, every time the nitrogen base is attached to the first carbon, every time the nitrogen base is attached to the first carbon of the pentose sugar, first carbon of the pentose sugar, every time. Okay. Now moving ahead, talking about the phosphate ion. So this phosphate ion, okay, either in the form of phosphoric acid or in the ionized form as shown over here, this phosphate ion or the phosphoric acid, the phosphate ion is attached to the fifth carbon of the sugar. So if this is the structure of the sugar molecule, okay, and this is the fifth carbon, CH2OH, right? And this one is the first carbon, this one is the second, this one is the third, this one is the fourth, and this one is the fifth carbon, right? See, this structure, this numbering is important to understand the structure of the nucleic acid. So every time the phosphate is attached to this fifth carbon, okay, with the help of the ester bond, with the help of a ester bond, okay. So when this sugar, it is attached with the nitrogenous base, then this unit is known as the nucleoside. Now this unit is known as the nucleoside. When the sugar contains the when the sugar contains what, the nitrogenous base attached to its first carbon, then it is known as the nucleoside. And when the phosphate ion is also attached along with the nitrogenous bases, when the phosphate ion is also attached, okay, now what happens here? When the phosphate ion is also attached, now this unit is known as nucleotide. Okay guys, so this is the structure of nucleic acid. Now I have included a table. Why? Because this is really important. In your NCERT, diagram, the chemical formula of these nucleoside and nucleotide are mentioned. And we will be conducting the NCERT session also in the end. Okay. Now what happens? Here, there are certain bases, like the purine bases are the adenine and the guanine, and the pyrimidine are the cytosine, thymine, and uracil, as told you. Now, the nucleoside, that means the nitrogen bases along with the sugar. So, it always ends with sine. You can see when adenine combines with the sugar molecule, it forms adenosine. Okay, when the sugar is ribose, it forms adenosine. When the sugar is deoxyribose, it forms deoxyadenosine. Okay, similarly about the guanine, guanosine and deoxyguanosine. Similarly, see here about the thymine and the uracil. As I already told you that uracil is only found in case of RNA. So uracil forms uridine and thymine in case of DNA forms thymidine or deoxythymidine. See, it is not specifically required to mention deoxythymidine in case of thymine. Why? Because thymine is specifically found only in case of what? Specifically found in case of DNA. Now talking about the nucleotide, that means when the phosphate ion is attached. So now it becomes, adenylate, deoxyadenylate, similarly thymidylate or uridylate. Okay, and depending upon the nitrogen base is present, the nucleic acid will be formed. Okay guys, so now let us discuss the type of the nitrogen bases. Okay, now let us discuss the type of the nucleic acid. So first one is the DNA, deoxyribonucleic acid. Okay, deoxyribonucleic acid, the genetic material which is found in our cell. Okay, deoxyribonucleic acid. Now the DNA molecule, it is a polymer of nucleotide, and this DNA molecule is a double stranded structure. We know that Watson and Crick, they gave us the double helical model of DNA, and we'll be discussing the double helical model of DNA as well. So according to the double helical structure, the DNA molecule, the nucleotides in the DNA, they are connected with the help of phosphodiester bonds, and the phosphate which is present at the fifth carbon here, okay, in the one nucleotide. If this is first nucleotide here, right? So here is at the fifth carbon the phosphate ion is attached. Here at the fifth carbon the phosphate ion is attached. In the next nucleotide, what happens? The phosphate will be attaching to the third OH. Here at the third position the OH was present, remember, and at the fifth position to the carbon phosphate is attached. Now this phosphate will be binding at the third OH position, okay, with the help of the phosphodiester bond, and this is how the polymer of nucleic acid is formed. Okay, this is how the polymer of nucleic acid is formed. So in a polynucleotide chain, different nucleotides bind together by phosphodiester bonds. Okay, the phosphate attached to OH at C5 position, at carbon 5 position, reacts with the OH group at the C3 position, that means the carbon 3 position, and thus the linkage is called 3,5 phosphodiester bond. Okay, now as I told you that DNA molecule is a double helical structure. Okay, so both these strands of DNA are anti-parallel in nature. That means the direction of the phosphodiester bond in both the strands of DNA are anti-parallel to each other. In the one strand, the phosphodiester bond is forming in the 3,5 direction, whereas in the other strand, the phosphodiester bond will be formed in the 5,3 direction. Okay, so this is how the the DNA molecule is formed. Now, DNA is a double helix and it takes a turn at every 10 base pairs. It takes a turn at every 10 base pairs. That means, DNA, it behaves like a ladder. It behaves like a ladder. Okay. And, the spacing between the two nitrogen bases, for example, if this is the nitrogen base and it is present like this. So, the spacing between the two pairs of nitrogen base is 0.34 nanometer. So the spacing, for example, here you can see the structure. This is, here A is bonding, double bond with thymine, and C is forming triple bond with guanosine. So in between this base pair and in between this A double bond T, the gap here is 0.34 nanometer. Right? And after every 10 base pairs, the DNA molecule is taking a turn. Okay, so now if we have to calculate the pitch of the DNA helix, so the pitch becomes 0.34 into 10, that means the 3.4 nanometer is the pitch of the DNA molecule. Okay, and talking about the diameter, that means the the distance from the phosphate to the phosphate molecule, distance from the phosphate of one strand to the phosphate of another strand. So the diameter of the DNA molecule is 2 nanometer or 20 angstrom. It is the same thing, right? So it is 2 nanometer or 20 angstrom. Okay, guys. Now what happens while when the DNA molecule is folding, it is revolving. It is a sort of a revolving structure. So what happens? Groove-like structure appears in the DNA. Okay. So there are two types of grooves. One is the major groove, another one is the minor groove. And these grooves are important from the functional point of view. The grooves are important in the attachment of DNA binding proteins. And these DNA binding proteins are involved in the process of replication, okay, replication and transcription. Okay, so those who still do not know what is a double helix, so when DNA molecule, see, DNA is a polymer of nucleotide, and what is nucleotide? So nucleotide is formed by like this a structure. Okay, here at this carbon phosphate is present and here is the nitrogen base. So this is a typical structure of a nucleotide, and when these nucleotide they combine together, okay, so this is one chain, and on the opposite side in front of this chain, what happens? Again, another chain of the DNA molecule is present, like this, but the direction of the phosphodiester bond formation is opposite. So now two strands are present, and these two strands are connected with the help of hydrogen bonds. Okay, so these two strands are present but in opposite direction, so anti-parallel direction. So these are the two strands of DNA. Okay, these are the two strands of DNA. Now, as I was discussing about the grooves, the eukaryotic DNA, it contains the histone protein. During the folding of the DNA, the histone protein binds to this major groove of DNA. The histone protein binds to the major groove of the DNA. Okay, the major groove is this one, where maximum distance is found between the backbones of the DNA, right? So this was about the, the grooves which are found in the DNA, and they're important from the functional point of view. Okay. Moving ahead, let me tell you some really important property of DNA. See, this DNA molecule is a double helical structure, as you can see over here. But when you treat this DNA molecule under high temperature, or you put, it in a heating condition, so what happens? Both the two strands of DNA, they starts to unwind. That means they starts to separate with each other. They starts to separate or unwind. Okay. Why? Because of the temperature, because of the high temperature condition. Okay. And this property, if you continue to provide heat condition to the DNA molecule, what happens? Ultimately, both these strands of the DNA will separate, and this phenomena of separation of both these strands of the DNA is denaturation of DNA. This phenomena is denaturation of DNA. What happened? Both these strands of DNA got separated. So this is one chain of nucleotides, this is another chain of nucleotides, and both these nucleotide chains are arranged, anti-parallel to each other in the formation of a DNA double helix. But what happens upon application of high temperature? Both these strands, they separate, and this phenomena is denaturation of DNA. Okay. Now what happens if you cool them? Okay, slowly or gradually. So what happens? Again, both these strands will combine. Again, both these strands will combine with the formation of the hydrogen bonds between the nitrogenous bases, and this phenomena is known as renaturation of DNA. Now this phenomena is known as what? Renaturation of DNA. Okay. But what if you try to cool it suddenly? If you try to cool it suddenly, okay, now what happens? If there is sudden temperature change from very high temperature, what happened? Very low temperature is provided. Now what happens? The quenching of DNA molecules takes place. That means they stay there in their denatured form forever. So DNA is denatured forever. Okay, when sudden change in temperature is provided. Okay, so this is DNA quenching. Okay. Now let us discuss about another type of nucleic acid, which is the ribonucleic acid or the RNA. Okay. So RNA molecules are generally present in the single stranded form, and as I told you that the nitrogen bases which are found here are all same except for the uracil, because the uracil, in place of uracil in DNA, thymine is present, but here uracil is present. Okay, so the four types of nitrogen bases are adenine, guanine, cytosine, and uracil. Okay, and the structures are also shown over here. Now let us know what are the different types of RNA available and their structures also. Okay, so see, the RNA can be classified into two broad categories: the genomic RNA and the non-genomic RNA. When RNA is present in the genomic RNA form, that means they are behaving as the genetic material. They are behaving as the genetic material. Okay, but it is very rare in nature. There are few examples like the reovirus or the wound tumor virus, like the reovirus, okay, or the wound tumor virus. Okay, here what happens? Here the RNA is behaving as the genetic material. Okay, but now talking about the non-genomic RNA. So these non-genomic RNA are basically divided into two broad categories. One is the mRNA or the messenger RNA. Okay, another one is the functional RNA. These functional RNAs includes transfer RNA or tRNA, ribosomal RNA or rRNA, small nuclear RNA or snRNA. These are the functional RNAs which are involved in various functions in the cell, and we know the messenger RNA or mRNA is involved in the transformation of the information. We know that DNA contains the genetic information, and it passes those genetic information to the RNA molecule, and specifically to this mRNA molecule. And with the help of the information which is present in the mRNA molecule, now the protein synthesis takes place. Okay, so this is how the cellular information travels, okay, from DNA to mRNA to the protein. Okay, so mRNA is involved in the process of protein synthesis. So now let us study the very first type of RNA, which is the ribosomal RNA. I hope each and every point is clear. Okay, so the ribosomal RNA. Okay, we know that ribosome, we have already studied in chapter cell, that ribosome consists of two subunits. Remember, one is the larger subunit, and another one was the smaller subunit. One was the larger subunit and another one was the smaller subunits. See, the ribosome is made up of rRNA along with proteins, rRNA along with proteins, and we have already studied about these two structures. So now I'm going to tell you the type of rRNA molecule which are involved in the formation of ribosome. So the function of ribosomal RNA or rRNA is the formation of ribosomes. Okay, so they form the major component of the ribosome. Okay, and they are the insoluble RNA, and the majority of the RNA which is found in the cell is the rRNA. So 80% of total cell RNA is what? rRNA. Okay, and they are synthesized in the nucleolus region. Remember, when I took the class of the chromosome, I told you that the satellite chromosomes and the secondary, I told you about the secondary construction also, and I told you that the DNA which was present in the secondary construction region is rDNA. That means it contains the information for the synthesis of rRNA, and all those regions were combined together to form a structure which was nucleolus. Remember? Okay, so this is about that particular structure only. You can relate both the classes, because both the in both the classes we have discussed about a common thing. So now when we say that the rRNA synthesis takes place in the nucleolus, we clearly understand each and every point. But there is an exception, this 5s rRNA. But first let me tell you the different types of rRNA involved in the formation of ribosomes. Okay, first let me tell you different types of rRNA which is involved in the formation of ribosomes. See, if this is the two subunits of the ribosome, okay, so we know that ribosomes are the structures which are found in organelles which are found in both prokaryotes as well as eukaryotes. In prokaryotes, 70s ribosomes, and in eukaryotes, 80s ribosomes are formed. So this one is the larger subunit, and below smaller one is the smaller subunit. So the 70s subunit of the prokaryote, it is made up of 23s rRNA and 5s rRNA. Whereas the 80s subunit of ribosome is made up of 28s rRNA, 5.8s rRNA and 5s rRNA. Talking about the smaller subunit in case of prokaryotes is made up of 16s rRNA, whereas here in eukaryotes it is made up of 18s rRNA. What is S? S is the Swedberg unit. S is what? S is Swedberg unit. It is a indirect measurement of density and size of the molecule. It is a sedimentation coefficient. Okay, so these are the different types of rRNA molecules which are involved in the formation of the ribosomes. When we talk about the prokaryotes, so each and every type of rRNA is formed in the cytoplasm itself because there are no membrane-bound organelles found. But when we discuss about the eukaryotes, we know that there are well-formed membrane-bound structures. And in case of eukaryotes, all the type of ribosomal RNA are formed or synthesized in the nucleolus except the 5s rRNA. This 5s rRNA is synthesized inside the nucleus but outside the nucleolus. It is synthesized inside the nucleus but outside the nucleolus. Okay. Now, what is the function? So, first of all, the rRNA are involved in the formation of ribosome. They are also involved in providing the attachment site to the mRNA and tRNA during the process of translation. That means during the process of protein synthesis. Okay. During the process of protein synthesis, what happens? First of all, mRNA molecules attach to the smaller subunit of the ribosome. So that means it is providing attachment site to the mRNA molecule. Then the tRNA molecules is also attaching to the ribosomal unit. Okay, so it is providing attachment site, and also the larger subunit of the ribosome, it contains the ribozyme activity. That means the enzymatic activity. Peptidyl transferase enzyme is the ribozyme. Okay, peptidyl transferase enzyme is the ribozyme. This is a really important point because normally whatever enzymes we study are normally protein in nature. They're normally proteinaceous. But this peptidyl transferase enzyme, which is involved in the process of peptide bond formation during the process of protein synthesis, is an example of ribozyme. That means here the RNA molecule is behaving as an enzyme. Okay, and this activity is due to the larger subunit 23s rRNA. Okay, now see, as I told you that there are different types of RNA molecules. I hope this structure is visible. This is 70s, this is 80s. In 70s, the larger subunit of 70s is made up of 23s type of rRNA and 5s, and the smaller subunit is made up of 16s rRNA. Similarly, in the eukaryotes, it is made up of 28s, 5.8s and 5s, and the smaller subunit is made up of 18s rRNA. right? Now, as I told you that in case of prokaryotes, since there are no membrane bound cell organelles, so each and every type of RNA molecule will be synthesized in the cytoplasm itself. But in case of eukaryotes, since we know that there is a well developed nucleus present, and the synthesis of RNA takes place in the nucleolus, okay, in the nucleolus region, except the 5s rRNA. This 5s rRNA is synthesized, although it is synthesized inside the nucleus only. Inside the nucleus only. Why? Because students, DNA is present inside the nucleus, and the information for the synthesis of RNA molecule comes from the DNA. So it is synthesized inside the nucleus, but outside the nucleolar region. Okay, so the 5s rRNA is the exception. Okay, so this is the first type of rRNA, which is the ribosomal RNA. Now talking towards the second type, which is the messenger RNA. So this messenger RNA or mRNA, term messenger RNA was given by Jacob and Monat. Okay, and this messenger RNA is the informational RNA. Why? Because it carries the information which it has received from the DNA molecule, and then according to the information received, it is involved in the process of protein synthesis. That is why it is the informational molecule. And once the process of protein synthesis is done, once the protein is synthesized, this mRNA molecule, it dissolves. Okay, it disappears. It do not exist. And it is 1 to 5% of total cell RNA. That means it is present in the minimum amount in a cell. It is present in the minimum amount in a cell, and it is involved in the process of protein synthesis mainly. Okay, the mRNA, it contains the information in the form of triplets. And these triplets are known as the codons. Okay, and by reading these codons, these triplet information, the sequence of the amino acid is decided during the process of protein synthesis. Okay. Moving ahead towards the next type of rRNA, the functional RNA, which is the tRNA, or also known as the transfer RNA. From the structure point of view, it has the most complex sort of a structure. See, tRNA is the smallest RNA. It is the smallest RNA because it is made up of only 77 to around 207 nucleotides. Okay, and talking about its abundance, so it is present approximately in 10 to 15% of total cell RNA. Okay, so in maximum amount, which RNA is present? rRNA is present. Next to rRNA is the tRNA, and at the last is the mRNA in the minimum amount. Okay, approximately 80% was the ribosomal RNA, approximately 10 to 15% is the tRNA, and mRNA is approximately 1 to 5% only. Okay, now this this tRNA is the soluble RNA. Okay, and what is the function of the tRNA? Why is it termed as transfer RNA? Because they carry amino acid to the ribosome during translation. Translation is the process of protein synthesis. Okay, so which amino acid needs to be brought is present, the information is present in the mRNA molecule. But how does the amino acid comes during the process of protein synthesis, and how does they combine? So transfer RNA acts as a mediator. Transfer RNA reads the information which is present in the mRNA molecule with the help of its anticodon loop. And then accordingly, depending upon the type of the amino acid codon present, the tRNA brings the amino acid. And this is how the peptide chain is formed. Okay, and sometimes the tRNA, it does contain some unusual nitrogen bases. See, we know that in case of RNA, adenine, guanine, cytosine, and uracil, these are the 4 type of nitrogen bases which needs to be present. But sometimes the tRNA molecule, not all the RNA, this tRNA molecule, it contains some unusual nitrogen bases like the pseudouridine, inosine, dihydroxyuridine, or methylated guanosine. Okay, so these are what? These are some unusual nitrogen bases which are found sometimes in the structure of tRNA. Although the RNAs are present in single stranded form, but sometimes what happens, the nitrogen bases, they form pairing, or they form bonds within that single strand itself, depending upon the complementarity of the sequence. As you can see, this is a tRNA molecule, this colorful balls which is shown over here. Okay, so this is a tRNA molecule, but you can see at certain regions, the bonds are formed here. At certain region, you can see triple bonds are present. At certain regions, double bonds are present. So these bonds represent the bonding between the nitrogen bases of the single strand only. Okay, so within that particular strand, sometimes this tRNA molecule forms bond, and it resemble, or it looks like a double stranded molecule, but it is exactly a single stranded molecule. Okay. Now see what happens at 5 prime end, at its 5 prime end. See here, this is the 5 prime end. Okay. The repetitive sequence of guanine nitrogen base is present. Repetitive sequence of guanine nitrogen base is present. Okay. Then what happened? At the 3 prime end, ACC is present. At the 3 prime end, this sequence ACC is present. Okay, so at the 5 prime end, guanine is repeatedly present, and at the 3 prime end, ACC is repeatedly present. Okay, yes, Shubhankar. Yes, and I have answered your doubt also. You asked me that the 5s rRNA, and I have repeated that point already. I have already repeated that point, and now I'm discussing the structure of the tRNA. In case if you have further doubts, do let me know. I'll be explaining them. Okay. So now, talking about the structure of the tRNA. Okay. So this tRNA has a complex structure, and its 2D model, which is also known as the clover leaf model, because it resembles in a structure to the clover leaf. Okay, was proposed by Hawley, and this structure is really important. You will be studying about all these information in class 12th once again when you'll be studying molecular basis of inheritance. Okay, but this is something really basic, and I want you to know it from right now when you are studying biomolecules for the first time. Okay, so see, according to the cloverleaf model, which was proposed by Hawley, the 2D model of the tRNA, the tRNA molecule has certain loop-like regions. See, you can see these loop-like regions. Okay, these loop-like regions are present because in these loop-like places, unusual nitrogen bases are present. So there are total 4 loops: first, second, third, and fourth. Okay, loop 1 is known as DHU loop. Loop 1 is known as DHU loop, and this DHU stands for dihydroxyuridine. I told you dihydroxyuridine is an unusual nitrogen base. So DHU loop stands for dihydroxyuridine, and with the help of this DHU loop, the tRNA is able to recognize the amino acid. So what happens during the process of protein synthesis? See what happens during the process of protein synthesis. If we need methionine amino acid, we have read about the different types of amino acid, right? And now we need methionine amino acid, for example. So how does the tRNA molecule brings? First of all, each and every amino acid has their own amino acyl synthetase enzyme. Okay, has their own amino acyl synthetase enzyme. That means if methionine is needed, so methionine also contain amino acyl synthetase enzyme. So first of all, what happens? This tRNA identifies their amino acyl synthetase enzyme with the help of this DHU loop. Okay, loop 2 is the anticodon loop. That means the anticodon is the complementary sequence of the codon. Codon is present in the mRNA. This is the mRNA molecule, right? And here is the codon. Okay, and the anticodon or the NODOC, anticodon or also known as the NODOC, is the complementary of the codon, which is present in the tRNA molecule. So tRNA molecule reads the sequence of the mRNA with the help of this anticodon loop. Okay, now this extra arm, that is loop 3, is also present, but its functional is quite unknown. Okay, the loop 4, which is known as the ribosomal binding loop, with the help of this loop 4, what happens? This tRNA molecule it attaches or it binds to the ribosome. Okay, it binds to the ribosome when it brings the amino acid. Okay, and this is how the tRNA molecule is helping in the process of protein synthesis. So there are multiple loops present. Okay, now apart from these RNA, mRNA, rRNA or tRNA, there are certain special type of RNAs which are also found in the cell. The name of those are snRNA, also known as the small nuclear RNA, hnRNA, also known as the heterogeneous nuclear RNA, or miRNA, or also known as the microRNA. Okay, this snRNA or small nuclear RNA, they are involved during the process of splicing. What is splicing? What happens during the process of eukaryotic RNA synthesis? When the RNA synthesis is taking place in the eukaryotes, the eukaryotic RNA contains some functional as well as the non-functional region. Some functional, and this zigzag is the non-functional region. Okay, so this non-functional region needs to be removed from the functional part. So what happens? Certain proteins and rRNA and snRNA molecules, they combine together to form a spliceosome complex. Okay, and these RNA molecules and the protein molecules, with the help of these structures, the non-functional region is removed from the newly formed RNA and the functional part is joined together, and this is the mRNA. This is the fully processed mRNA. Okay, the newly formed RNA, which is just synthesized from the DNA, which contains both the functional and the non-functional part, it is known as the hnRNA. It is known as what? The hnRNA. See, it is written over here, heterogeneous nuclear RNA or pre-mRNA. Okay, and the miRNA is what? So miRNA are sort of regulatory RNA. A sort of what? Regulatory RNA. So it is synthesized in eukaryotic cell to regulate the expression of RNA. If protein synthesis is not needed, so what happens? This miRNA is synthesized and it binds to the mRNA molecule. This is how the protein synthesis is prevented. So this is used in the regulating of the mechanism. Okay, so these are the other special type of RNA molecule. Now, how does this RNA synthesis takes place in the cell? So as I told you that first of all, the copying of DNA molecule. So formation of DNA from DNA is DNA replication. Right? And it takes place with the help of DNA polymerase. DNA replication takes place with the help of DNA polymerase. Right? The process of formation of RNA from DNA is known as transcription. It is known as what? It is known as transcription, and transcription takes place with the help of RNA polymerase or transcriptase enzyme. Okay, so in eukaryotes, what happened? There are three different types of RNA polymerase are found. And these three different RNA polymerase are involved in different functions. Like RNA polymerase 1 synthesizes rRNA. All the ribosomal RNA are synthesized with the help of RNA polymerase 1 except the 5S rRNA. Similarly, RNA polymerase 2, it synthesizes mRNA. Okay, and that means the hnRNA also, because hnRNA is what? It is the pre-mRNA. RNA polymerase 3, it synthesizes the tRNA molecule and also the 5S rRNA, which was not synthesized by RNA pol 1, and the snRNA. So it is synthesized by RNA polymerase 3. This is how the synthesis of RNA takes place inside the cell, and these are the different types of RNA molecule which are found in the cell. So now we have a clear-cut understanding of the nucleic acid which is found in the cell, DNA and RNA, and also of the components from which they are made up of: nitrogen bases or the nitrogenous bases, sugar, and the phosphate ion. And we also know the positions where they are attached. Okay. Now let us go through the this NCRT part. See, the Taq polymerase, it is not tag, it is Taq polymerase. You will be studying when we will be discussing the PCR in the biotechnology part. So in the polymerase chain reaction, this Taq polymerase helps in synthesizing the new DNA strands. Okay, so Taq polymerase is different. Okay, now see, nitrogen bases. Okay, so nitrogen bases, for example, adenine is the representative of purine. You can see two cycles are present, and pyrimidine is single cycle. Uracil is present over here. So nitrogen bases, and when they combine with sugar molecule, as you can see here, it is the ribose sugar. Okay, it is the ribose sugar here. Okay, in both the places. So what happens when with the ribose sugar, adenine is combined, name becomes adenosine. When the uracil is combined, the name becomes uridine. And when the nucleotide is formed, that means the phosphate is also attached to the fifth carbon, as you can see over here. Now the term is known as adenylic acid. So nucleotide ends with acid. Okay, it is adenylic acid. Or if it was uridine, so it was uridylic acid. Okay, so this is how you can identify the nucleosides, nucleotide, and nucleic acids. Okay, now let us see what is written over here. So nucleic acids, the other type of macromolecule that one would find in the acid insoluble fraction or the retentate, remember? Okay, in the retentate part of any living tissue is the nucleic acid. So these are polynucleotides, that means these are the polymer of nucleotides. Together with polysaccharides and polypeptides, these comprise the true macromolecular fraction of any living tissue or cell. That means these nucleic acids, along with the polysaccharides and the polypeptides, that means the proteins, they together forms the actual content of the macromolecules which are found in the cell. Because we know that lipid is not chemically macromolecule. Okay, because the molecular weight of lipid do not exceeds 800 Dalton. Right? It was present in the acid insoluble fraction because of its non-polar nature. Now, for nucleic acids, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is a monosaccharide, that means the sugar, pentose sugar, and third is a phosphoric acid or phosphate. Okay, as you notice in figure 9.1, the heterocyclic compounds in nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purines while the rest are the substituted pyrimidines. Right? The skeleton heterocyclic ring is called either ribose, okay, or deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid, while that which contains ribose is called as ribonucleic acid. Okay, that is what we have discussed right now. Now, the nature of bond linking monomers in a polymer. Since we have discussed the type of bonds in individual categories itself, but your NCRT, it contains a separate paragraph where it is discussing about the nature of the bonds. So I'll be reading each and every line, although I have already discussed about these points in individual parts. So let us see what is written over here. In a polypeptide or a protein, amino acids are linked by peptide bond, which is formed when the carboxyl group of one amino acid reacts with the amino group of the next amino acid with the elimination of a water moiety. And this process is called dehydration because water molecule is moved. Okay. In a polysaccharide, the individual monosaccharides are linked by a glycosidic bond. I told you, right? In the formation of a polysaccharide, glycosidic bond is formed. In the formation of protein, peptide bond is formed. Okay, this bond is also formed by the process of dehydration. That means here also a water molecule is eliminated. Now this bond is formed between two carbon atoms of two adjacent monosaccharides. Okay, that means the two carbon atoms which are present adjacent to each other, they will be combining to form this polysaccharide, this glycosidic bond like this. Okay, and one carbon is present outside the ring. Now, in a nucleic acid, a phosphate moiety links the third carbon of one sugar of one nucleotide to the fifth carbon of the sugar of the succeeding nucleotide, that means the next one. And this is how the phosphodiester bond is formed. The bond between the phosphate and the hydroxyl group, the bond between the phosphate and the hydroxyl group of sugar is an ester bond. So, phosphate is forming two ester bond. One, when it is attached to the fifth carbon, then also it is forming a ester bond. And when the polymer is formed, that means it is again attaching to the third carbon, then it is also again forming a ester bond, and that is why we call it as phosphodiester bond. Okay, so as there is one such ester bond on either side, therefore it is called as phosphodiester bond. Now, nucleic acids exhibit a wide variety of secondary structures. So this is an important point, right? Nucleic acid exhibits a wide variety of secondary structure. For example, one of the secondary structures exhibited by DNA is the famous Watson-Crick model. This model says that DNA exists as a double helix. The two strands of polynucleotides are anti-parallel. Okay, the direction of one strand is from 5' to the 3'. The direction of another strand is from, just anti-parallel. In front of the 5', 3' is present. Okay, that is run in the opposite direction. The backbone is formed by the sugar phosphate sugar chain, sugar phosphate sugar chain, and the nitrogen bases are projected more or less perpendicular to the backbone. That means if the sugar, if this is the sugar and this is the phosphate attached, okay, so the nitrogen base, it is present at 90-degree angle to the backbone. If this is the backbone of the nucleic acid, so nitrogen base is projected at a 90-degree angle, okay, but face inside. Now A and G of one strand compulsorily base pairs with T and C respectively. That means A binds with T with the help of double bond and G binds with C with the help of a triple bond. Okay. There are 2 hydrogen bonds between A and T and 3 between G and C. Each strands appear like a helical staircase. सीढ़ियां देखिए ना, helical staircase. Now each step of ascent, this is an important point, is represented by a pair of base. That means each step is a pair of base. For example, this is A double bond T, this is C triple bond G, right? Then at each step of ascent, the strand turns 36 degree. That means at each step the strand is turning or rotating with the degree 36 degree angle. With the 36 degree angle the DNA strand is rotating, right? One full turn of a helical strand would involve 10 steps or 10 base pairs. Okay, now attempt drawing a line diagram. Now if you try to draw the line diagram, okay, this is the first base pair, this is the second, this is the third one, this is the fourth one. So what happens? The pitch would be 34 angstrom. I told you how. Okay, 34 angstrom or 3.4 nanometer, it is same thing, right? Then the rise per base pair would be 3.4 angstrom or 0.34 nanometer. This form of DNA is the bDNA. This form of DNA which we have discussed right now is bDNA. We know that there are other forms of DNA also, aDNA, bDNA, cDNA, ddNA or zDNA also. So you'll be studying about these different types of DNA in class 12th. The most common type of DNA is the bDNA, which was discussed by Watson and Crick. Okay. See, in higher classes, you will be told that there are more than a dozen forms of DNA named after English alphabets. I told you, right? With unique special features. Okay. So the property which we have studied right now is about the bDNA. Okay. So this was for the nucleic acid session, students. In tomorrow's class, we will be discussing enzymes and then we will be completing this chapter. We have already discussed all the 4 different types of biomolecules: carbohydrates, proteins, lipids, and nucleic acids. Although I understand that the nucleic acid which is covered in this session includes some part from class 12th also, but this is for your betterment because you'll be able to solve questions. Okay, now see what happens. Since this channel, the name of this channel is Let's Crack NEET UG in English, therefore the lectures here are available in English. Okay, throughout in English. Okay, that's the restriction here. So let me tell you my timings once again. I'm live daily at 5 p.m. I'm live daily at 5 p.m. with brand new lectures, and tomorrow I'm going to tell you about enzymes, the most important part. Okay, I'm also live daily at 9 p.m. with practice sessions. Okay, every day we pick any one chapter either from class 11th or class 12th and we solve questions, okay, at 9 p.m. So today topic of our practice session is ecosystem, class 12 topic, right? And on Unacademy learning app, my timing is 11 a.m. in the morning. You can follow me on Unacademy learning app using the link provided in the description box to get all the updates regarding my class timings. Okay, students. So if you have still not subscribed for Unacademy platform, either Unacademy Plus or Iconic, so do not delay and do not forget to use my referral code, which is AG001, to get an instant 10% discount and get a chance to enroll in my personal batch. Okay, so this is my referral code AG001. And in case if you have not pressed the like button and if you have not subscribed to this channel, so do not forget to press the like button and subscribe and share. Okay, so that's it for today, students. Thank you.